Hello, welcome everybody. My name is Zafer Yüksel. I'm a medical doctor specialized in medical genetics, and today I would like to share you the mitochondrial disease testing in a clinical practice presentation. And I would like to mention about myself shortly. I studied at School of Medicine in Eskişehir Osman Gazi University, Turkey. I did my 4-year residency training at Medical Genetics Department at the same university, also my hometown, and my thesis was related with CNVs in autism cohort. After this period, I went to Yale University for further research in autism population and using SNP arrays and vast technologies. I collaborated with researchers who were leading Mendelian genomic project, which is still ongoing in its second 4-year phase. After the Yale period, I worked almost for 2 years as a resident physician in Women and Children Hospital in Mersin, Turkey. I have clinical genetic experience over 5 years, mainly focused on rare disorders, and besides, I have deep insight and hands-on experience for cytogenetics, molecular genetics, and cell culture. I have started to work for Centogene in May 2016, and I'm evaluating RES, VGS data, as well as mitochondrial analysis, analyzing genetic data obtained by different methods such as Sanger sequencing, MLPA, and qPCR, preparing medical reports for the genetic findings, and advising clinicians in case of specific medical questions. Well, I would like to mention about my presentation today. So here's the plan. I'm going to introduce you first the mitochondrial disorders and its core point, the respiratory chain, and common clinical features of mitochondrial disorders and the mitochondrial disorders at a glance, inheritance pattern for mitochondrial disorders, and how to make a diagnostic approach and how we are doing here at Centogene, and the molecular genetic tests offered by Centogene here, and also I will give some examples from our cases that we diagnosed here. Okay, so mitochondrial disorders are a group of related clinically diverse genetic diseases. Its effect mainly seen on high energy consuming tissues and organ systems. Because mitochondria is the main place where the aerobic metabolism occurs. Prevalence is found for mitochondrial disorders in developed countries changed, but as mentioned in several different reports from countries like England, Sweden, Finland, and Australia, it is like 1 in 5,000 and 1 in 8,500. And the inheritance of the mitochondrial disorders depends on where the gene is located on. It's either mitochondrial genome, mitochondrial DNA, or it's the nuclear genome. So let's have a look to the respiratory chain. The respiratory chain is located within inner mitochondrial membrane, which is between the intermembrane space and the matrix of the mitochondria. As you see, there are 5 complexes in total, and mitochondrial genome actually contributes to encoding all of them, and except complex 2. Because complex 2 is succinate dehydrogenase, and the related gene is located on nuclear genome. So as you see about the numbers, it's interesting that 37 Gs contributes for the encoding of the several proteins. For complex 1, 7 proteins. For complex 3, 1 protein, cytochrome B. For complex 4, 3 proteins. And for complex 5, mainly ATPases, 2 proteins. And as you can see, a main burden is for the nuclear DNA here, because more than 1,000 genes are just defined that they contribute for the mitochondrial function and its maintenance. Also, I wanted to mention here about the shortly about mitochondrial DNA and nuclear DNA comparison, the protein number that is encoded, and the number of genes that are responsible for this issue. Well, the mitochondrial chain is very important because it's the essential final common pathway for aerobic metabolism. Therefore, tissues and organs that are highly dependent on aerobic metabolism are having more numbers of mitochondria. For example, while erythrocytes are having almost no mitochondria, and liver cells can have up to 2,000 mitochondria per cell. That is why these organs in the picture preferentially involved in mitochondrial disorders, and thus we have such a distribution of clinical features. For example, when we start on the upper right hand, like the eye, we can have like ptosis, external ophthalmoplegia, optic atrophy, pigmental retinopathy, and when you check about the skeletal muscle, proximal myopathy and exercise intolerance can be the clinical features for the mitochondrial disorder. And when you check about with the heart, cardiomyopathies or conduction disorders. For inner ear, it can be sensorineural deafness. For pancreas, it could be diabetes mellitus. And the main clinical feature usually accumulated in central nervous system, and these features are like fluctuating encephalopathy, seizures, dementia, migraine, stroke-like episodes, ataxia, and spasticity. I would also like to mention about that these features can present at any age, and these kind of features could be affecting multiple organs at the same time or just the onset change. One organ might be involved in earlier times and the other organ might be involved in later times. I would also like to mention, and because this feature is mainly goes unrecognized, a high incidence of mid and late pregnancy loss as a common occurrence for mitochondrial disorders. That should be also kept in mind when we are considering about mitochondrial disorders. Here in this list, you can find not all the, but very well-known examples of the mitochondrial disorders at a glance. And you can see, for example, some classifications that we can make among mitochondrial disorders as mitochondrial DNA deletion syndromes and ataxia neuropathy syndromes. Yeah, and here you can see the list of the well-known ones. Well, the most important thing is how these disorders are being inherited. Let's start with the genes located in mitochondrial genome. The way that mitochondria are passed on is quite unique. Both egg and the sperm contain mitochondria, but following the fertilization of the egg, the mitochondria from the sperm are destroyed. This means that only the mother's mitochondrial DNA is passed on to the next generation. So even if a man has a mutation in his mitochondrial DNA, it will not be passed on. In contrast, a woman with a mutation can pass it on all to her children without any gender discrimination. Although there is only one nucleus per cell containing one set of nuclear DNA, here depicted in the pictures with the 2 chromosomes, cells contain hundreds of mitochondria, and each mitochondrion contains several copies of mitochondrial DNA. Therefore, we have to talk about several factors to consider inheritance patterns, particularly for maternal inheritance. And we have 3 terms here, like homoplasmy, heteroplasmy, and the threshold effect. Homoplasmy is like each human cell contains thousands of copies of mitochondrial DNA, and at birth, these are usually all identical. So the situation is called as homoplasmy. By contrast, individuals with mitochondrial disorders resulting from mutation of mitochondrial DNA may harbor a mixture of mutated and wild-type mitochondrial DNA within each cell. So this condition is called heteroplasmy. So in the picture, you can see that the mutated DNA is shown as red dots. So the wild-type mitochondrial DNA is shown as its green dots. So you can see the heteroplasmy among the cells. And there's also the threshold effect, because there will be some kind of a proportion between those mutated DNAs and wild-type mitochondrial DNAs. So the proportion of mutated mitochondrial DNA might exceed a critical threshold level before a cell expresses a biochemical abnormality of the mitochondrial respiratory chain. The percentage level of mutated mitochondrial DNA may vary among individuals within the same family, and also among organs and tissues within the same individual. This is one explanation for the varied clinical phenotypes seen in individuals with disorders caused by mutation of mitochondrial DNA. Here we see the other inheritance patterns that would be also possible for mitochondrial disorders. And here we have autosomal dominant and autosomal recessive inheritance pattern due to the genes carried on chromosome from 1 to 22, and X-linked inheritance patterns due to the genes located on X chromosome. And you see here the difference between X-linked inheritance and maternal inheritance. In maternal inheritance, dependent for mitochondrial DNA, it shows no gender discrimination. Mother can inherit the mutated mitochondrial DNA to sons and daughters. But in X-linked inheritance, usually females are carriers. They could be affected, but usually milder than males, and usually males are affected. Well, we talk about mitochondrial DNA and its inheritance issues, but what about nuclear DNA? And as we mentioned that almost more than 1,000 genes involved about the mitochondrial protein coding, maintenance, and so on. Here you can find the roles that nuclear DNA is playing for mitochondria. So like nuclear genetic disorders of the mitochondrial respiratory chain can be listed as 3 subclass, like because nuclear DNA is encoding structural subunits and assembly factors and the translation factors. And also nuclear DNA is associated with mitochondrial disorders due to the mitochondrial DNA depletion and deletions, because mitochondrial DNA replication and maintenance is maintained by nuclear DNA, and also nuclear supply and balance and mitochondrial dynamics and biogenesis. Also, Q enzyme 10 biosynthesis is an important issue here I want to mention separately. Well, after talking about mitochondrial function, at a glance mitochondrial disorders and their common clinical features and inheritance patterns, so how we shall approach when we have the suspicion for mitochondrial disorder? Actually, we should focus on specific findings, characteristic features. As a clinician, also by myself, we shall ask these questions if the clinical picture is a progressive disorder. It's very important. And if the clinical picture having complex central nervous system features or single neurologic symptom and another system involvement. Maybe the most striking one, children with lactic acidosis. And very important also the family history, if just supporting any kind of inheritance patterns, particularly the maternal inheritance. Or the other, of course, autosomal recessive or autosomal dominant inheritance patterns. Okay, we had the suspicion that it can be a mitochondrial disorder. Then comes another important step, if it is a specific clinical picture or it's a highly suggestive picture. Because here in this picture, if you see the outer circle of this picture, there are clinical informations. Maybe they are not visible, but please visit our website and check under the mitochondrial genetic testing tab. So you can find a detailed and high resolution picture of this one. So here, it's not always easy to just find specific features for each mitochondrial disorders, but at least you can try to get some kind of clue, because clinical variability is quite high and varied presentations even within the family is possible. At the end, many individuals do not fall into one specific disease category, yet it's still a good alternative approach to find at least one or two specific findings. Here I would like to share another approach published by Morova and friends in 2016, and this diagnostic criteria is quite useful because it gives scores according to three main categories, and which are like clinical signs and symptoms, which you can get a maximum 7 points, metabolic and imaging studies, which you can give maximum 4 points, and the morphology studies, which you can give maximum 4 points. And there are also other specific symptoms having high points, and I would like to mention because they're quite suggestive for any mitochondrial disorder, like ophthalmoplegia, elevated plasma lactate levels, elevated cerebrospinal fluid lactate levels, and alanine levels also, and urinary titratable acidity excretion. Leigh syndrome specific MRI findings, those would be summarized as striatal necrosis or striatal hyperintensities, which is mainly like focused on ganglionic level. And also there are some other issues like abnormal mitochondria finding with electron microscopy or SDH positive blood vessels. These ones also should be taken into consideration. And also there are other findings if you find them in higher percentage, and those could be also ranked with higher points, like red fibers and COX negative fibers, reduced COX staining, and so on. And if you have the score 1 after applying this diagnostic criteria, it's unlikely a mitochondrial disorder. If you have the score between 2 and 4, then it's possible that it might be a mitochondrial disorder. If you have a score between 5 and 7, it's probable. And if you have a score between 8 and 12, it's definitive mitochondrial disorder. So this approach is quite handy, and I would suggest that to use during daily clinical practice. Here, if you would just like to summary about this diagnostic criteria, the clinical picture is very, very important because all starts here. All starts from bedside and then it goes to bench side. So clinical picture, I would like to highlight again all these specific symptoms and the distribution of all these cases. And of course, also after having clinical investigations and physical examinations, there are some investigations that will be helpful to just give the decision about diagnosis and to choose the appropriate testing later on. And I guess the most easy ones are plasma lactate measurements and cerebellar MRI findings among them. But of course, for sure, normal results for plasma or CSF results does not exclude the mitochondrial disorders, but they are very helpful. So here you see the MRI, MRS, and CV, EMG, and EAG investigations, which are very helpful at the second tier as metabolic and imaging studies. And if we go to the morphology studies, here you can find some examples from the third category, like histopathology. So in this picture, it demonstrates red fibers, which is typical for MERV syndrome. And also, if it's possible from muscle biopsies, you can investigate by electron microscopy, and this is here an electron microscopy picture, which is showing an enlarged and walled mitochondrion in the middle side of the picture here. And also, if it's possible, it would be very helpful, not only just by diagnosing, but also directing the NGS results. When you have respiratory chain enzymology results, and think about later on, we will also talk if you have a unknown significant variant, so you can assess better and you can evaluate better NGS report. Well, the most important question, when do we have to go for molecular genetic testing? So molecular genetic testing can be done when you have suspicious and having at least non-invasive supportive investigation like MRI or minimal invasive investigation like plasma lactate levels. Also, some of the invasive yet uncommon tests can support the found variants and boost the interpretation of molecular tests, particularly respiratory chain enzymology is very helpful about this one. One other important issue is the choice of the sample for molecular genetic testing. Mutant mitochondrial DNA is often undetectable in blood cells, and this necessitates examination of muscle or other affected tissues. But on the other hand, for example, in Pearson syndrome, mitochondrial DNA deletions are usually more abundant in blood than in other tissues. And in progressive external ophthalmoplegia, for example, mitochondrial DNA deletions are confined to skeletal muscle. So for these cases, muscle biopsy is a better choice for molecular genetic testing. For other disorders, affected tissue should be sampled as well. For example, this could be blood usually, and second, muscle, but this could be other disorders as well. I mean, other organs as well, like liver, the third tier maybe. And in theory, molecular genetic test approach can be categorized like into three, like serial testing of single genes and multi-gene panel testing, like simultaneous testing of multiple genes, and genomic testing for the entire genome, like entire mitochondrial genome, or whole exome sequencing, or whole genome sequencing. Okay, so I would like to share our approach here at Centogene. And when we here have a suggestive symptoms and supportive family history and supportive investigations that I mentioned, like MRI or plasma lactate levels, simply and initially, and then we think about that this could be a mitochondrial disorder. And then the second step arises. If it's a specific clinical picture or it's a highly suggestive clinical picture. If it's a specific clinical picture, here in Centogene, we offer different panels that is covering all the related genes, and they can have a better approach and much more quicker results than the other approach. So here I would like to mention about some of them, like Leigh syndrome and mitochondrial encephalopathy panel, progressive external ophthalmoplegia panel, Leber optic atrophy panel, and myopathy rhabdomyolysis panel. And also there's another issue I would like to mention here in this slide. There are some genotype-phenotype correlations. So for example, if you have a case and you made a muscle biopsy and you just saw that red fibers and you think that, oh, it's very specific clinical picture, and I think that would be a MERV syndrome. So if you check these two variants that I mentioned in the table that you can see in the tRNA genes, for MERV syndrome, these two variants can explain almost up to 90% of the cases. So that would be also a good choice to just go with these molecular tests when you have a specific clinical picture. Also, this table is very helpful. For example, also under the rRNA genes, if you have aminoglycoside-induced non-syndromic deafness, this is one variant. You can just check this one hotspot, and then you can diagnose. So this is the choice for the specific clinical picture if you have. But if you don't have, or your specific clinical picture tests resulted negative, then here we go with the Cento mitogenome. And Cento mitogenome, I'm going to give more information later on, just covering all the mitochondrial genome, and also we can check large deletions in mitochondrial DNA. If this is also negative, we can go with the two methods. One is Cento-Mito comprehensive. This is our mitochondrial panel, and it includes Cento mitogenome, like covering all the large deletions and SNVs in mitochondrial DNA, and also the nuclear genes that are related with the mitochondrial disorders. Also, there's another choice, whole exome sequencing. That's also a good choice. And after, if you have negative results, of course, you can choose whole genome sequencing. And when you have a positive result, carrier testing for family members, prenatal and PGD tests, and also genetic counseling should be addressed. Here, I would like to start with Cento Mito Comprehensive. Actually, naming is like this. I would like to clarify this one. We have two different panels. One is covering the other one. Actually, the smaller one is Cento Mito Genome, which is covering the entire mitochondrial genome, 100% coverage at 1,000 reads. And mitochondrial heteroplasmy can be confidently detected by this panel, and also large mitochondrial gene deletions can be detected by this panel. So there's another panel that covers Cento Mito Genome, which is called Cento Mito Comprehensive. So this comprehensive panel includes 374 nuclear genes related to mitochondrial disorders, and mean coverage for this panel is 120 reads. And more than 94% of the bases of those nuclear genes are covered at least more than 20 reads. So also in comprehensive, as it's including Cento Mito Genome, it's also possible to detect large mitochondrial gene deletions. I would like to mention about the different test methods and the disorders that we detected here in Centogene. For example, Cento Mito Genome that can detect SNVs and CNVs in mitochondrial genome detected a deletion in a case, and we diagnosed the case as Pearson syndrome. And there was another case also which we detected and diagnosed as Leigh syndrome, and this syndrome couldn't detect any variants by West, and it revealed no relevant phenotypic information. But when we checked with Cento Mito Genome, we could detect the single nucleotide variant in mitochondrial DNA. So this is the advantage of Cento Mito Genome, because it has 100% coverage of mitochondrial DNA and at least 1,000 reads. Also, you see the other examples that we made diagnoses like 3-methylchromatyl-CoA carboxylase 2 deficiency and thymine metabolism dysfunction syndrome 2, which is also diagnosed together with the whole exome sequencing. And I would like to mention with our cases. Here we had a case which we performed Cento Mito Genome panel, and the case had autism, cerebellar hypoplasia, motor delay, delayed speech and language development, ataxia, and microcephaly. So for this case, the chromosomal microarray was negative, and the whole exome sequencing couldn't reveal any variant that would be related with a clinical phenotype. And then, as Cento Mito Genome panel can cover the single nucleotide variants and copy number variations in mitochondrial DNA, so when we performed the analysis for this case, we detected a single nucleotide variation in ATPase6 gene. And so it made the diagnosis certain as Leigh syndrome. And we reported this variant according to the current nomenclature. This was the case actually I shared also in the slides of mitochondrial statistics. And there was another case, and this case also had a request for Cento Mito Genome panel, and this case had failure to thrive, pancytopenia, hepatomegaly, elevated liver function tests, lactic acidosis, striatal necrosis, and developmental regression. Here, striatal necrosis is a very important, a very nice finding. It should be kept in mind for any mitochondrial disorders. It's quite specific. And then when we perform the test also, because as it covers the SNVs and CNVs, we detected for this case a deletion that encompassing mitochondrial genes like NDL4, ND4, and ND5 genes. And here you can see the orange PCR picture. Here, the B and the C fragment is showing here that the deletions encompassing these 3 genes, and it's like a 3.4 kilobase long deletion. So that the case was diagnosed as Pearson-Marrow pancreas syndrome. These were the cases that diagnosed in Centogene. Here I would like to share my references, and thank you very much for your interest. And if you would like to have any questions, please just call us or email us. So we would like to answer or give more information about our mitochondrial panels, which are called like Cento Mito Genome Panel and Cento Mito Comprehensive Panel. Thank you very much for your interest, and I would like to answer the questions also if you have any. Yeah, and here we have a question that you mentioned in the table that there are over 1,000 genes located on the nuclear genome, and we have 374 genes in the Cento Mito Comprehensive. What is the reason for this? Actually, the important thing is about the relation with the phenotype, with the mitochondrial disorders, because there are genes that are found to be, responsible for encoding or maintaining the mitochondrial function, but they're still not related with the mitochondrial disorder phenotypes. But perhaps in the future, perhaps in the near future, they will be correlated with the genes, and so that we will be able to also include them in our panels. And we have another question. What about the saliva sample for the testing? Is it a good idea to study with saliva sample if we couldn't collect blood or tissue? If we really couldn't collect blood or tissue, at least if we have this option, we can go with this one. We have the chance to detect, but the preference should be the blood and the affected tissue. But of course, in clinical practice, it happens, or it was just possible to have saliva sample. So yeah, of course, we can go with the saliva sample as well. Yeah, thank you very much. And I guess that's all. We don't have any more questions. Thank you very much for your attendance. Hope to see you later with other presentations. So bye for now.